Welcome dear students. Today we are going to learn this MSc first year CHI-230 course, that is Bioinorganic Chemistry. Today here we are going to learn some related or remaining topics which is related to the hemoglobin. We already discussed about the hemoglobin, their structure, their mechanism, okay, and also there are different factor, that is pH factor and the pressure, osmotic pressure, like this factor which is affect the affinity of the oxygen to bind the hemoglobin. Today we are going to learn, that is what the role of hemoglobin in the red blood cell. Okay, here, so let's start with this topic. In the 1 microliter of the blood which is present in the adult human, they have the RBCs about 4 lakh to the 6 lakh. They contains the WBC, that is 4,000 to the 11,000, and the platelets are 1,50,000 to the 4 lakh. The each human red blood cell, that is RBC, contain approximately 270 millions of the hemoglobin molecule, and each hemoglobin molecule carrying the 4 heme group, and this 4 heme group contains what? The 4 iron, that is 4 Fe molecules. Okay, 4 Fe molecules. That here what I am saying, the RBCs, this contains about 270 millions of the hemoglobin. In our RBC contains what? 4 lakh to red blood cell. How many red blood cell present in our body? 4,000, 10,000, 40 lakh to the 60 lakh. Okay, and each RBC contains approximately 270 millions of the hemoglobin, and each hemoglobin contains what? 4 heme group, and each heme group contains 1 Fe. This 4 heme group contains 4 Fe. Okay, then hemoglobin comprises about a third of the total RBC volume. The red blood cell of the average adult human male store collectively about 2.5 gram of the iron. Okay, they has to store 2.5 gram of the iron. You know how iron is important for our body. Okay, here that means we require the maximum amount of the iron to prepare the hemoglobin. And here, what is the function of this hemoglobin? This hemoglobin is the oxygen carrier in our body. What is the function of the oxygen? Oxygen convert the carbohydrates, what we take, the food, into the energy. Okay, that's why this hemoglobin is very, very important. And this hemoglobin structure possible only due to the ion, that is Fe, which bind the porphyrin ring. Then only they can do their function properly. Okay, the cell which is developed, the cell, that is RBCs, that cell which are developed in the bone marrow, where the cell RBC is developed, which is developed in the bone marrow, that is the stem cell, and the circulate for about 100 to 120 days in the body before their component are recycled. Each circulation take about 20 seconds. Approximately 1/4 of the cell in the human body are what? Red blood cell. Okay, that means in our body the maximum percentage of the red blood cells are present. The mammalian red blood cell are unique among the vertebrates as they don't have a cell nucleus. Okay, that means in the red blood cell they don't have the nucleus. You can see this is the red blood cell, and this cell doesn't have the nucleus. Okay, in their mature form. So there is no chromosomes or the DNA are present in the cell. If you consider the cell, they contains the nucleus, and at the nucleus there is a DNA and the chromosomes are present. But in the RBCs, there is a no any nucleus. That means there is a no any chromosomes and the DNA are present in that cell. This cell have the nuclei in the early stage of the development, but this is excluded as they mature in order to provide more space for the hemoglobin. That means in the early stage, this cell which has the nucleus, but whenever the cell get mature, then this nucleus get ruptured, exclude for what? For providing the space for the hemoglobin. Provide the space for hemoglobin. This cell also does not have a cellular organelles such as their mitochondria. Okay, here this RBC strange shape is what? Biconcave disc. You can see this is the structure of the RBC. This having the structure of what? The biconcave disc that is found, that is round and the flat. This RBCs has no any nucleus. The nucleus is excluded from the cell as it matures, and the RBCs can change their shape to and the amazing extent without breaking, and it can be squeezed single file through the capillary. That's why here they can be squeezed a single file through the capillaries. And RBCs contain, this RBC contain what? This RBC contain the hemoglobin. Okay, here these are some hemoglobin, structure of the hemoglobins. This hemoglobin unit are present in per RBC. Each RBC contains hemoglobin unit, and this hemoglobin, that means in one RBC there are the 4 hem group are present. The each hemoglobin contain what? 4 hem group, and each hem group contains what? 2 alpha amino acids and the 2 beta units, and here they contain the active sites of the hem unit, that is contain the iron, that is Fe. This contain the Fe. Okay, this is about the red blood cell. Let me see the type of the hemoglobin. There are mainly the 3 types of hemoglobins are present. First, that is hemoglobin A. This is called as a Fa. Second type is the hemoglobin B. Sorry, this is hemoglobin HB, hemoglobin A. Second type is hemoglobin F, and the third type is hemoglobin S. There are mainly 3 types of hemoglobins are there. Here, first let me see the hemoglobin A. From the structural point, this is the hemoglobin A which is present in the adult, and this hemoglobin F which is present in the fetal, that means newborn baby. The hemoglobin F type which is present in what? The baby, newborn baby, and this hemoglobin A which is present in the adult. Okay, here this is the structure of the hemoglobin A, that is adult hemoglobin. This is called as a adult hemoglobin, that is hemoglobin A, and this is the structure of what? Hemoglobin F, which is present in the fetal. Fetal means the newborn baby, hemoglobin F. What is the difference of that? From the structural point of view, the adult hemoglobin which is composed of 4 hem group. Here it contains 2 alpha and 2 beta structure. In the adult hemoglobin, that is hemoglobin A, contains the 4 hem group. In that they contain the 2 alpha structure unit and the 2 beta units. That is called as a hemoglobin A. Here, in case of hemoglobin F, that is a fetal hemoglobin, this beta structure is replaced by the delta units. That means it contains 2 alpha units and 2 delta units. That is called as a hemoglobin F. This is very important for the MCQ pattern and also in the next examination. The hemoglobin A contains what? 2 alpha and 2 beta units, and hemoglobin F contains 2 alpha and 2 delta units. Okay, the delta chain is a 72% identical in amino acid sequence with the beta change. Here what happened in the change is substitution. What happened here? The substitution of serine residue for the histidine 143 in the beta change. Beta change contain histidine which can be replaced by the serine. This is the histidine which is present in the hemoglobin A, but this is replaced by the serine group. Okay, that's why this is this present in the hemoglobin F and this is present in the hemoglobin A, that is in adult, and this is in the fetal. In addition, the fetal hemoglobin and the adult hemoglobin are found to be a different near the 2,3-BPG binding site. The 2,3-BPG binds less tightly with the deoxy form of the fetal hemoglobin as compared to the deoxy form of the hemoglobin. Okay, the babies are born with the hemoglobin F. Okay, here whenever the newborn baby is born, that time they contains what? The hemoglobin F, that is called as a fetal. But after a few months, the body shut off its synthesis and start to make the hemoglobin A. That is, whenever this baby which is moved to becomes the adult, then the hemoglobin from the body is going to changes from hemoglobin F to the hemoglobin A. That is called as a hemoglobin switch. Okay, that means when a baby is moved from fetal to the adult state, that time their hemoglobin F is shut off or removed, and production of this hemoglobin F is going to stop, switched, and the production of hemoglobin A is produced. Okay, start, and that's why this is called as hemoglobin switched. Okay, this is the structure of histidine. What is the difference of that? Here in hemoglobin A, it contains the beta chain. Beta chain contains histidine. This is replaced by the delta chain, and which contains what? Serine unit. This is also important for the MCQ point of view. In the F16, in hemoglobin F, it contains delta chain, and this delta chain contains serine. In hemoglobin A, it contains beta chain which contains histidine unit. Okay, this hemoglobin A is called as adult hemoglobin. Hemoglobin F is called as fetal hemoglobin. Okay, next is the organic compound, that is a BPG, that is 2,3-biphosphogyl binds to the hemoglobin A and reduces its oxygen affinity by the factor of 26. This at the first instance will make one wonder. Why? Interestingly, this increase in the oxygen binding affinity of the fetal hemoglobin, that is hemoglobin F, relative to that of the maternal, that is hemoglobin A. This difference in oxygen affinity allow the oxygen to be effectively transferred from maternal to the fetal red cell. The transfer of oxygen from what? Mother to the fetus, or mother to the baby. Okay. Next type, that is hemoglobin S, that is also called as a sickle cell anemia. Okay, this is the anemia. What happened here? This is the normal shape of the hemoglobin, but their shape of, this is the normal structure of the RBC, but here this normal shape of the RBC or red blood cell is going to change into the sickle shape. That's why this is called the sickle red blood cell, or this is also called as a sickle cell anemia. What happened here? The sickle cell anemia is caused by the mutation in the beta globin chain. What happened here? There is a mutation and change in the beta globin. Okay, there are the two chain is present in the adult hemoglobin, alpha and beta. There is a mutation in alpha, there is a mutation in the beta chain of the hemoglobin, which causing the hydrophilic amino acid glutamate, which can be changes to the hydrophobic amino acid valine. That means what happened in the beta chain? It contains the hydrophobic glutamic acid, and this glutamic acid is changes to the hydrophobic, that is water hating amino acid valine. Okay, that means there is a change in the hemoglobin beta from hydrophilic, that is water loving glutamic acid, this is amino acid, to the hydrophobic, that is water hating amino acid, that is valine. Due to that change, the shape of the normal red blood cell is going to change to the sickle shape. That's why this is called as the sickle red blood cell. Why this happen? Due to the malaria. In area where the malaria is a problem, the people's chances of survival actually increase if they carry sickle cell carrier. The malaria parasites has a complex life cycle and spends part of it in a red blood cell. In a carrier, the presence of the malaria parasite which causes the red blood cell with defective hemoglobin to rupture prematurely, making the plasmodium unable to reproduce. The polymerization of the hemoglobin S affect the ability of the parasite to digest the hemoglobin. It digests the hemoglobin. What is the signs and symptom? This is not feeling good, headache, fever, low blood count. These are the some sign and the symptom for what? Hemoglobin S. This is also called as a sickle cell anemia. What happened is, here this is the structure of what? The normal adult hemoglobin. Okay, this is HA. Here this amino acid in the nucleotide contains leucine, taurine. This is the glutamic acid. Okay, this glutamic acid which is changes to what? Valine. You can see here, this is the HBB sequence in the mutant adult hemoglobin. This glutamic acid get changes to the valine, and due to that change, this shape of RBC is going to change. It look like a sickle. That's why this is called the sickle cell. This RBC with the deoxy HBS changes its disc shape to the sickle shape. The valine unit make the HBS unit to stick together. That means to polymerizes, forming the strand and changes the shape of the RBC. They change the shape of the RBC. Okay, this is about the sickle cell anemia. Thank you for watching this lecture. Here we completed our topic, that is what? Hemoglobin. Next time we will learn the next topic which is related to the biomolecules. Okay, so thank you very much for watching this lecture. Watch this lecture which is very beneficial for you. Thank you very much.